So now we are going into understand the clinical research introduction was completed. So now we will understand how the phases, on what phases it has to occur clinical research. As per sir's experience, there are 4 phases, and what phases and what dates and what timings you need to be carry on this clinical research. Sir will be explaining about the phases of clinical research.
Yeah. Now we will discuss about the parts, the flow of clinical research, how clinical research of a drug, device or a procedure will occur. Okay. The first one is, as you know, medical research. Medical research is on human beings, right? Medical research before human beings. Okay, this is clinical research. Now this clinical research is divided into 2 parts. One is preclinical research and clinical research. These are the 2 major phases, and each one will have different phases and with different purpose. We will look into it. The first one is, right, preclinical research. Again, preclinical research is divided into 3 phases. One is in silico, in vitro, in vivo. Okay, we will check what is in silico, in vitro, in vivo. The first part of preclinical research is in silico. What do we do in silico? What is meant by in silico? Silico means computer. First, a device or a drug or a surgical procedure has to be designed in the computer, right? You have to use graphics, or you have to do the research on existing literature. Whatever you do, first you do basic research. All that comes under in silico. Once you have got all the knowledge, all the information, existing information, then you move on to animal trials. In animal trials, there are 2 parts. One is in vitro and in vivo. In vitro means taking an animal part like this and applying the drug or a device or a technique which is applicable. For example, you are working on a drug which reduces the stomach pain, maybe stomach. What do? You take the gut of a rabbit or gut of a rat. You put the gut of a rat into a petri dish and you induce constriction, right? Peripheral constriction. Or what do? You stimulate the intestine so that it will contract and produce pain. Now you take the drug and put the drug here. You will check whether the intestine is relaxed or not. Is your drug is relaxing the intestine or not? So basically you are taking the part of the animal out and doing research on it. That is considered as in vitro testing. Once this is done, once you find that this drug is having some effect on the intestine of the rat, then what do? You go for in vivo test. In vivo, what do? You take a rat or an animal, you induce severe abdomen pain or cramps into it through using other drugs. Then you inject the test drug. Once you inject the test drug, whether the rat is feeling relaxed or not. So after in vitro, you have to check in vivo. Is it working on the in vivo or not? If you do on the live animals like this, this completes the animal testing. So again, preclinical phase is divided into in silico and animal testing. Animal testing, in vitro and in vivo. After you complete this, you get safety data, you get efficiency data. Means what is the safer drug? If this is the weight of the animal, right? If for this weight of the animal, this drug is safer, this amount of drug is safer, then human being is this weight. What is the safest drug? You decide through the existing knowledge and existing literature, what is the safe drug for human beings, safe dose for human beings, and also is it efficient in human beings or not? You are going to test in the clinical research.
So basically, sir, here what I understood, the dosage will be understood exactly, according to the weight, how much dosage or what we call as mg, how much it is needed, and how this reaction is going into the animal. So based on that, then it will be applicable in the human life as well.
Right.
So not like this is a caution. I think the more of over aggravations, what we call, that can be avoided due to this preclinical, at the same time in the 3 phase of steps. So this is a good idea. This can be implemented in homeopathy in further future as well.
Yeah, definitely. 3 important things we want from clinical research is whether the medicine is working or not. That is the first one. Second one is safety. In safety, what do? What is the safe dose for human beings? That we have to get. Efficiency and safety. You take any experiment in clinical trials, you test only 2 things. One is efficiency, one is safety. Here efficiency, how we check whether it is relaxing the abdomen, relaxing the intestine or not. That is efficiency. and what is the safe dose for human beings, that is safety,
Yes.
right? Here we are using the animals and considering the safety for human beings, not for the animals. Okay. Yeah. Once we complete the preclinical trials, then we move on to clinical trials. Clinical research, it is on human beings. It is carried in 4 phases: phase 1, phase 2, phase 3, phase 4. And we will check what is the importance of each phase. Why do we need to divide into 4 phases? The important rule here is phase 1 is the first phase. The medicine is coming from animals to the human beings for the first time. What we have to do? We have to test in minimal number of patients, healthy patients. Okay. What do we do in phase 1? The main objective, see, it will have two objectives: safety and efficiency. But main objective of phase 1 is safety first, whether the drug is safe or not. That is the first one. On an average, it is done on 8 people. Okay, and these are only healthy people. It is not done in the diseased people. These are the 3 points in phase 1. Phase 1 trial is completed. Now we have checked the safety, whether the drug is safe for human beings or not, whether it is, we will also check efficiency, if the drug is efficient or not. If safety and efficiency is proved in phase 1, then the drug moves into phase 2. Okay.
So as you said here, healthy human beings.
Yeah.
So how you consider he is healthy, and before coming into the clinical research as a participant, what kind of instructions you give them and what kind of tests they have to be done? So based on that, the efficacy is more of related, no, sir. So that you explain, sir, what tests to be done, how you consider a healthy human being, and how you consider diseased human being. Once if you give a gist of idea, so they'll get an idea about it. What is healthy exactly?
Right. In clinical research, you take any phase 1, phase 2, phase 4. There is not phase 4. Phase 4 is different. We will look into it. Phase 1, phase 2, phase 3. There are 2 criteria: inclusion criteria and exclusion criteria. These criteria should be satisfied. Inclusion criteria should be satisfied. Exclusion criteria should be ruled out. What it means is, the protocol will say and the regulatory authority will say, if you want to take the people, they should have these characters, right? Inclusion characters. If they have these characters, they are eligible. That is, inclusion criteria should be satisfied. And they will also say beside that, exclusion criteria, if they have these problems, they are to be excluded. Means exclusion criteria should be ruled out. They should not have the things which are mentioned in the exclusion criteria. They should have the things mentioned in the inclusion criteria. If inclusion criteria is satisfied and exclusion criteria is ruled out, then only you take the patient to the clinical trial. What that inclusion criteria, exclusion criteria depends on the particular drug, device or procedure. For example, it is a HIV drug. So what criteria? You cannot do the HIV testing on a healthy human being, right? You have to do directly take the diseased human being. All those things are mentioned in that. Maybe for example, if you are doing a drug and on fever. So normally fever drugs acts on liver and kidneys. We have to check whether the patient is having good liver function or good kidney function. It depends. In clinical trials, everything is based on the situation. So for this particular drug or for this particular device or for this particular procedure, what is the inclusion criteria, what is the exclusion criteria, depends on many features, right? It is a ocean, and there are some specialized clinical research organization who do phase 1 clinical trials, and it is a multi-billion-dollar business. Okay, so they are strictly guided, especially in phase 1 is very strictly guided. Even other phases also, but most strictly regulated phase is phase 1.
Yeah, continue.
Yeah, we go to phase 2. We got safety data. Once in phase 1 it is proved the drug is safe, or the dosage of the drug which is safer is proved, then we take to phase 2. In phase 2, we consider only diseased people. For example, if it is a fever, we take only fever patients. If it is a diabetes, we take only diabetic people. Here the inclusion criteria and exclusion criteria is different. Here the inclusion criteria says the patient should have diabetes, and exclusion criteria says he should not have liver disease, something like that. The important thing, safety is always there. Here the importance is given to efficiency. What is efficiency here? Is the drug is efficient? Is the drug doing intended purpose? For example, if you are doing a drug on obesity, is the drug reducing obesity or not? That is done in phase 2. Here importance is always for safety, and also here efficiency is also considered. It is normally done around on around 80 people, because it is already safe, it is already done, so it is will be done on around 80 people. Once phase 2 is completed, we submitted the data to FDA or any regulatory authority. If it is satisfied, then it gives permission to phase 3. Of all the research, the major research is phase 3, and it is done on around 800 people. It is done. Here the thing you have to consider in phase 3 is, it is already safer, right? It is already efficient. How efficient is it, right? Is it efficient than the previous ones? You already have paracetamol for fever. Why regulatory authority is going to give permission if the drug is efficient, safer, but not safer than paracetamol? If it is safer than paracetamol, then only it gets permission. It is working, it is safe, but it is less efficient or less safer than paracetamol, they will not give permission. This is the race here. You have to always consider a drug, invent a drug, device or procedure which is safer and which is better than the previous one, then only the government will give permission. So here tested is a comparison, how safer it is compared to existing drugs, how efficient it is compared to existing drug, is done in the phase 3. If phase 3 is successful, if you can prove your drug, device or surgical procedure is better than the existing one, is safer than the existing ones, is efficient than the existing ones, then you move on to phase 4. Phase 4 is releasing into the market. You release the drug into the market. After phase 3, you get permission. The regulatory authority will give you permission. Yeah, this drug is good, you can sell in my country, so I will give you patent right for 8 years. Here important thing for homeopaths. Last month I have done a podcast with one of the senior guys, Dr. R. Vallavan, who is head of scientific research in Wilmer Schwabe. He said me important thing. Till now, homeopathy medicines did not have any patent right? For example, if you invent any new drug, if you prove any new drug, anyone can make it, right? There is no regulation on it. But since 2024, new regulations has come in homeopathy. If you invent a drug, you will be given exclusive patent rights for 5 years. You prove a drug and you will get exclusive rights for 5 years. Only you can make the drug for 5 years, and you can sell. No one can sell the drug. First time, that is the patent right is introduced into homeopathy. This is very good, because most of the pharmaceutical companies will now invest money so that they can make money. This is very good improvement in homeopathy. That this phase 4 already released into the market, and you just watch it. This is also known as post marketing surveillance. What is meant by post marketing surveillance? You will just watch the drug is safer, or if the drug is efficient than the previous one. If government thinks it is not safer, it will withdraw the drug. Phase 4 is for lifetime. Once you complete the phase 3, it goes into phase 4. Phase 4 will complete only when, if the regulatory authority decides that this medicine is not good. For example, we have nimucilide drug, right? It is withdrawn in very many countries. Means because there are many drugs now available which are more safer, more efficient than nimucilide. That's the reason the drug is withdrawn from many countries, but still available in India. Phase 4 ended means the drug is not available in the market. Okay, that is phase 4.
So as now, recently we have seen many things like in modern medicine, some cough syrups have been affecting some of the child of very 4, 5, 6, 7 years. So why that is happening, sir? What could be the reason? They don't follow all this phase 1, phase 2, phase 3, phase 4, or if they follow also, why these side effects now it is evolving? What could be the probable reasons, sir, according to you?
No, the recent case where there was a problem with cough syrup is not due to clinical research. It is clinical research is done. The drug is in phase 4. The problem is there was a defect in manufacturing. Wrong product or wrong additive or some reaction has occurred in the medicine and it went to wrong. It is nothing to do with the clinical trials. It is to do with the manufacturing of the drug.
So how that we can avoid, sir, or in homeopathy also, as you said, the patent rights has been, so that could be the probable reason, I think. So how we can avoid this, sir, in coming future?
Yeah, very strict quality regulations. Allopathy medicines are strictly quality regulated, right? No one can make, there are a lot of things, but that strict guidelines are not there in homeopathy. The reason, that is a advantage of homeopathy, and you can say disadvantage also. How advantage? Because homeopathy doesn't have side effects, we no need to worry about that much, right? Anyone can make any drug and use any drug, because homeopathy medicines will not have side effects. The reason for not having side effects is we use drug in many very small quantity. For example, how small it is, Professor Ballare has identified it. For example, you take a paracetamol tablet, 500 mg paracetamol tablet, okay, small tablet. You cut the tablet into 1 crore pieces, right? 1 crore pieces, you cut the tablet. Take one small piece and put in 1 ml of liquid, right? What is the concentration? Same concentration we have in our dilutions. It is just few hundred or few thousand picograms per ml. It is coming to the same concentration, okay? That's the reason homeopaths need not worry about side effects. That is the advantage. What is the disadvantage? There is no regulation. You don't know whether you are using correct drug or not. You cannot test. There are no strict rules. Anyone can make homeopathic drug. So this is the disadvantage. So every system, every procedure will have advantage and disadvantage. This is the advantage we have, and that is the disadvantages we have as well. But this gap is closing now. That's the reason more homeopathic. You go to any conference, everyone is telling homeopathic MD, homeopathic pharmacy is the future of homeopathy, because we are going from unregulated procedures to, when we go to regulated procedures, we need more expertise, more people in that who can manage it.